differentiates between animal cells and plant cells. Component cells differentiates between animal cells and plant cells. Human beings are complex organisms, made of trillions of cells which vary in sheer size and structure the all organs into different forms and carry out specific functions. Improved technology has helped biologists to study the organism's cellular structure and foundation. They have even found ways to modify cellular structures and manipulate gene expressions for the enhancement of human health and environment. Robert Hooke discovered cells in the mid-17th century with their simple microscope and marked the genesis of an exciting era of cellular biology. A cell is the smallest unit of life and the building block of all living organisms. Understanding what makes up the cell and how a cell works is fundamental to all the biological sciences, such as biotechnology, genetic engineering, and molecular biology. So let's begin with whole organisms. And work down to the molecular level, organisms are made up of the organ system, such as the digestive system pictured here. Each of these organ systems are comprised of individual organs which work together in a coordinated manner. The organs such as liver, stomach, small and large intestines are a part of the digestive system. All these organs are made up of tissues. Tissues are groups of similar cells that perform the same functions such as the epithelial tissues and these muscle tissues pictured here. Tissues are made up of cells and these cells are the smallest units of life. Finally, cells contain organelles and molecules such as DNA.
The cells are classified into two types on the basis of the structure: prokaryotic cell and eukaryotic cell. Prokaryotic cells are the simple form of cells which make up unicellular organisms like bacteria and cyanobacteria. Prokaryotic cells, like membrane-bound nucleus and other membrane-bound organelles, the DNA, which is usually a single circular molecule found inside the cytoplasm of the cell. On the other hand, eukaryotic cells make up complex multicellular organisms like plants and animals. The eukaryotic cellular structure is more complex as pictured here. They have nucleus and other membrane-bound organelles. Animal cells are foundation of all animal. Let's examine an animal cell more closely. Animal cells are protected by a phospholipid layer that surrounds the cell and function as a gate that allows the entry and exit of molecules from the cell. In the center of the cell lies the nucleus. Nucleus acts as brain of the cell, aids in every metabolic activity of the cell. It's regulated by nucleus. It contains the DNA in form of chromatin and nucleus. Cells contain number of small, double-membrane, both organelle cell mitochondria.
Mitosis is the module about mitosis. There is a process of cell division which allow the regeneration of body parts to be turkey. Mitosis is the separation of the nuclear chromosome into nuclei identical through that are divided into four process main: propase, metapase, anapase, and temapase. An additional process called kinesis occurs after complete nuclear division. Before the cell they were to enter mitosis, I was going through a period of growth, and this ratings period is called interphase. At the interphase are of the cell gained material, interesting piece drawing with chromatin condenser and be sorted and struck to from chromosome. Each chromosome has two sister chromosomes joined together at the centromere of the nuclear, and the nuclear centromere separates, and the nuclear membrane going to bring do the centrosome. It with a pair of movable central support to opposite poles from a spindle bit we name matted during the microtubule madrapis. The spindle interact with the chromosome. This causes the chromosome to move in parallel to the cell division, or the chromatic accumulator plate sister have centromere inform the presence or the structure protein called kinerco hold the chromosome and adjust them to
Chromatin
the
spread at
analogous
the
spindle
kinetor
during
and their daughter
interphase.
chromosome,
Chromatic
along with the
spindle
differ
during
normally, move toward the opposite poles as the microtubule shorten the poles of the spindle. Also move apart and have to spread they move apart and help to spread the chromosome as they spread completely into two. Say an FH for an FS is considered complete telophase door. Telophase daughter chromosome arrive at the pole and the chromatin start to delete. Also, the nuclear report along with nucleol discharge there. The formation of the nuclear membrane kinesis. Kinesis is the parting of the plasma in animals for in the plasma membrane with the prints and the place their plasma into doctor called as quarantining. Another implants were missing and triggered at the center of the cell by the formation of a cell plate. The cell plate grows out dry till it needs the action while resulting in what Dr. Kill in the model you have learned. mitosis in the process of extraction of nuclear chromosomes. In total, it ensures the primary nucleotides and forward the primary plasmid metaphase and to the place improves the chromatin kindernis and becomes sour and tissue. From the chromosome bound to ground, microbullets of the spindle enter what the promising in anapsy. The sister chromatid spread out at the trough and move toward encephalitis. A microbial
Light and electron microscopes. The beauty of biology and all that we know today is due to the invention of microscopes. Microscopes had been available since the big beginning of the 17th century, but a lot of improvements were made in the quality of glass lenses. In the early 19th century, the branch of biology, dealing with microscope design, slide preparation, and examination is called cytology. Light microscopy. The light microscope uses light as a source of radiation and is shown as inside cells. It shows a simple structure like the nucleus, the cell wall, and the cell membrane. A light microscope is still used in basic studies in school today. Electron microscopy. Then come the electron microscopies, which made your microscopic were more detailed. Due to the electron microscopes, we not only show the cell wall, the nucleus, and the cytoplasm, but we also the more minute organelle, like the mitochondria, endoplasmic reticulum, and the Golgi apparatus. The light microscope were doing great, but no matter how much their design improved, there was an extent to which the light microscope could not show. Anything smaller than 200 nm could not be seen. Electrons are subatomic particles that have a negative electric charge, and they're found surrounding the nucleus of an atom called neutron. When a metal becomes very hot, some of its electrons gain so much energy that escape from their orbits. Like shooting stars, the energy in free electrons is associated with the proportion of waves. The higher the energy, the short wavelength. Electrons are very suitable from of radiation for a microscope for two major reasons. Firstly, the wavelength is extremely short. And secondly, because they are negative charge, they can be focused easily using electromagnetic. The magnet can be made to alter the path of the beam. Similar to how glass lens made to bend the light, there are two types of electron microscopes in use today: the transmission electron microscope (TEM) and the scanning electron microscope (SEM). In the transmission electron microscope, the beam of electrons is passed through this specimen before being viewed. Only those electrons that are transmitted are seen. This enables us to see different components inside the cells. In a scanning electron microscope, the electron beam is used to scan the surface of structure, and only the reflected piece is observed. The advantage of this is that surface structure, for example, like the chitinous outer body of insects, can be seen in great detail.